In 1953, the world witnessed a scientific revelation that would forever alter our understanding of life itself. This was not just another scientific discovery, it was a monumental leap forward, a moment that would be etched in the annals of history. This was the year James Watson and Francis Crick unveiled the structure of DNA, the molecule responsible for carrying genetic information from one generation to the next. Their groundbreaking work was built on the foundation laid by other scientists, including Rosalind Franklin, whose X-ray diffraction images were crucial in revealing the double helix structure. This elegant double helix, resembling a twisted ladder, holds the code that determines our traits, from the color of our eyes to our susceptibility to certain diseases. The discovery was akin to finding the key to a complex code, a code that had been written billions of years ago and passed down through countless generations. The discovery of DNA's structure was nothing short of revolutionary. It was a moment of triumph, a testament to human curiosity and perseverance. Scientists around the world were electrified by the news, realizing that they now had a powerful tool to unlock the mysteries of life. It opened up a whole new world of possibilities for understanding the mechanisms of life. Researchers could now explore how DNA replicates, how it mutates, and how it directs the synthesis of proteins, the building blocks of life. This newfound knowledge laid the groundwork for the field of molecular biology. Prior to this, the scientific community had only a rudimentary understanding of how traits were passed down from parents to offspring. The principles of inheritance, first outlined by Gregor Mendel in the 19th century, were well known, but the molecular basis of these principles remained a mystery. With the structure of DNA revealed, we finally had a blueprint to work with, a roadmap to decipher the language of life itself. Scientists could now begin to understand the genetic code, the sequences of nucleotides that make up our genes, and how these sequences are translated into the proteins that perform countless functions within our cells. The implications of this discovery were immediately apparent. It was clear that understanding DNA would have far-reaching consequences, not just for biology, but for medicine, agriculture, and many other fields. The ability to read and manipulate genetic information opened up new avenues for research and innovation. DNA, it turned out, was not some abstract concept, but a tangible molecule, a chemical entity that could be studied, manipulated, and understood. This realization transformed biology from a largely descriptive science into a precise experimental discipline. This realization set the stage for a new era in biology, one in which scientists would delve into the very essence of life, unraveling its secrets one base pair at a time. The study of DNA led to the development of powerful techniques such as DNA sequencing, which allows us to read the genetic code, and genetic engineering, which enables us to modify it. The discovery of the double helix structure marked the dawn of molecular biology, a field that continues to revolutionize our understanding of life and drive unprecedented advancements in medicine, agriculture, and biotechnology. From the development of life-saving drugs to the creation of genetically modified crops, the impact of this discovery is felt in countless aspects of our lives. It is no exaggeration to say that the unveiling of DNA structure was one of the most significant scientific achievements of the 20th century, paving the way for a future shaped by our growing knowledge of the very code that makes us who we are. As we continue to explore the depths of the genetic code, we are reminded of the profound impact that this discovery has had on our understanding of life and our ability to shape the world around us. The story of DNA's discovery is one of scientific collaboration, ingenuity, and a touch of controversy. It is a tale that spans decades and involves numerous brilliant minds, each contributing a piece to the puzzle that would eventually reveal the structure of life itself. At the heart of this story are James Watson and Francis Crick, two brilliant scientists working at the University of Cambridge in the early 1950s. Their partnership was marked by a relentless drive to uncover the secrets of DNA, a molecule that had already been identified as the carrier of genetic information, but whose structure remained elusive. Their quest to determine the structure of DNA was fueled by a shared passion for unraveling the mysteries of life. They were inspired by the work of other scientists and driven by a sense of competition, knowing that other researchers around the world were also racing to solve the same problem. Watson and Crick were not working in isolation. They were part of a vibrant scientific community that included many other researchers whose work was crucial to their success. Collaboration and communication were key elements in their journey as they exchanged ideas and data with colleagues both near and far. Their work built upon the research of many other scientists, including Rosalind Franklin, a skilled X-ray crystallographer. Franklin's expertise in X-ray diffraction techniques allowed her to capture images of DNA that were far more detailed than anything seen before. Franklin's meticulous work producing high-resolution images of DNA fibers proved instrumental in Watson and Crick's breakthrough. Her ability to produce clear and precise images of DNA allowed for a deeper understanding of its structure, providing the visual evidence needed to support theoretical models. Her famous Photo 51 in particular provided crucial evidence for the double helix structure. This image, with its distinctive X-shaped pattern, was a key piece of the puzzle that Watson and Crick needed to confirm their hypothesis about the helical nature of DNA. Using a combination of model building and careful analysis of existing data, including Franklin's X-ray diffraction images, Watson and Crick were able to deduce the now iconic double helix structure of DNA. They meticulously pieced together the information, constructing physical models to visualize how the components of DNA fit together. They envisioned two long strands of nucleotides, the building blocks of DNA, intertwined in a helical shape. This elegant structure not only explained how DNA could store genetic information, but also how it could be replicated and passed on from one generation to the next. This structure, they realized, elegantly explained how genetic information could be stored and transmitted. The double helix model showed how the sequence of nucleotides encoded genetic instructions, and how the complementary strands could separate and serve as templates for replication. The discovery of the double helix was a pivotal moment in the history of science. It marked the beginning of a new era in molecular biology, opening up new avenues of research and leading to countless discoveries about the nature of life and heredity. It catapulted Watson, Crick, and Franklin, whose contribution was tragically cut short by her untimely death, into the annals of scientific history. Their names became synonymous with one of the greatest scientific achievements of the 20th century. Their work laid the foundation for a new era of biological research, one that continues to yield profound insights into the nature of life itself. Today, scientists around the world build upon their legacy, using advanced technologies to explore the complexities of DNA and unlock the secrets of the genome. The DNA molecule, despite its profound implications, is remarkably simple in its composition. It is a long chain of smaller units, each one contributing to the overall structure and function of the molecule. This simplicity is deceptive, as it belies the incredible complexity and diversity of life that DNA is capable of encoding. It is made up of just 4 basic building blocks known as nucleotides. These nucleotides are the fundamental units of DNA, and they come together in various sequences to form the genetic instructions for all living organisms. These nucleotides, each consisting of a sugar molecule, a phosphate group, and one of 4 nitrogenous bases, are the letters in the language of life. The sugar and phosphate form the backbone of the DNA strand, while the nitrogenous bases extend from this backbone, ready to pair with bases on a complementary strand. The 4 nitrogenous bases, adenine, thymine, guanine, and cytosine, are the key to DNA's ability to store genetic information. Each base has a unique structure that allows it to form hydrogen bonds with a specific partner, ensuring the accuracy of genetic information transfer. These bases pair up in a highly specific manner. Adenine always pairs with thymine, and guanine always pairs with cytosine. This specificity is crucial for the replication of DNA, as it ensures that each new strand is an exact copy of the original. This predictable pairing, known as complementary base pairing, is crucial for DNA replication and gene expression. During replication, the DNA molecule unwinds, and each strand serves as a template for the formation of a new complementary strand. The sequence of these bases along the DNA strand is what determines the genetic code. This code is read in sets of 3 bases, known as codons, each of which specifies a particular amino acid, the building blocks of proteins. Just like the 26 letters of the alphabet can be arranged to create countless words and sentences, The 4 bases of DNA can be arranged in an almost infinite number of combinations, giving rise to the vast diversity of life on Earth. This genetic code is universal, shared by all living organisms, from the simplest bacteria to the most complex plants and animals. Understanding the structure and function of nucleotides is essential for comprehending the complexities of DNA. By studying these building blocks, scientists can unlock the secrets of genetic inheritance, disease mechanisms, and the evolutionary history of life. These simple building blocks are the foundation upon which the entire edifice of life is built. From the smallest microorganism to the largest mammal, all life forms rely on the precise arrangement of nucleotides in their DNA to function and thrive. They hold the key to our inheritance, our traits, and ultimately our very existence. By understanding DNA, we gain insight into the very essence of life itself and the intricate processes that govern our biological makeup.